of Arizona State. And what they were wanting to see in this second Science news story is if there's a bacterial infection, how does the sex life change of these fruit flies? So we've talked about this bacterium before, right? Wolbachia is something that has been studied for extremely long time now. In fact, if you take fruit flies from the wild and you try to publish a research paper, the bacteria consume the stuff that is otherwise used to make the drive reproduce. What do you mean, Wombat? For this situation, so Wombat, this is the first example of reproductive behavior looked at as a function of if the animals have been infected. Normally, the way this has been studied for Wolbachia is what happens to the animal's brains. So if you... We'll go ahead and pull up the old paper. So... this one. Yeah, here we go. So, Lara Gale, thank you for the lurking jerky, Lara Gale. Lara Gale, make sure to put the band hammer down on those smellies, Lara Gale. So, Wombat, this has been the context that Wolbachia has been usually looked at, is the fruit fly's brains. And what you'll see here is this first panel, there's these nice circular repeating size structure cells, and that's what you want in a brain, right? But then if you infect the animals with Wolbachia, this is a progression that you get. So everything starts to shrivel, fall apart, and you start getting actual physical holes in the brains of the animals. And so it is neurodegenerative in nature. And so the example, like, that's one of the reasons why anyone who's been doing any kind of study with Wolbachia and fruit flies, usually fruit flies in the wild are infected by this bacterium. It exists everywhere. And therefore, if you just bring them in from the from outside into the lab and do a behavior on them, you will get a question of, hey, are your flies affected by Wolbachia? Did you do a test and see if you had an infection or not? That's a fair ask, I think, right? So, but it turns out, here's the weird part, Wombat. So I mentioned that the flies are dramatically affected, right? If they have infected by the Wolbachia. And their reproduction rate is also changing. But here... So there's sex, we're going to go to the sex differences. It's different if a male is infected versus a female. This paper was the first time that they looked at what happened if a female was infected with Wolbachia. In this instance, Wombat, for a female, when infected, the infected females are more likely to mate more often and lay more eggs, and even want to reproduce and have special time with species that are not of their own. So, if anything, Wombat, this parasite, the Wolbachia, makes the fruit fly's inhibitions go away, basically, right? And want her to be more likely to have reproductive activity. Because then you're increasing the likelihood that there's going to be a spread of that bacterium as the animal is going to be reproducing or trying to at least reproduce. Does that make sense in terms of the directionality? Like, it makes sense in terms of what the parasite's drive would be, which is to maximize the output of individuals that it can spread to. But it's just very different than what it does from the male. Tested female, that hasn't been tested, Rose. That's a great question. Rose, the papers that have been looking at the male behaviors, they didn't give the males a choice between infected or uninfected females. They just gave them the females to mate with. And at least if you give them no choice, they mate at the same rate. That doesn't mean that they can't tell apart the differences. Maybe they can, maybe they can't, right, Rose? But they were at least mating at the same rate. If the males were infected, they would mate with an uninfected female just as often as an infected one. If they mate with an uninfected, no, that's the catch, Sparky. The female will not become infected from the male. The way they do get infected, though, Spartacus, it's much more aggressive and delicious from their poos, Sparky Bugwash. So if they defecate in a given area where everyone else is eating, what ends up happening then is that's how they're acquiring the bacteria. There's other parasites that they acquire the same way. Another one called the trypanosome. That's a noodle-looking parasite as well. That's also acquired. They're just eating their feces pretty much because they're pooping on where they live, and that's what's putting it into newer animals as well. So let's say that these uninfected animals lay embryos, and the embryos hatch to be larvae. Well, now if some males come over, or infected females, and just poop on the food that the larvae are eating, they're more likely to pick it up, right? And that's how it can spread throughout the population. And then it's also going from infected mom to embryo to the next generation too. That's another way that it's going down to. Is that when these animals are infected with Wolbachia, male reproductive behavior has been studied. And in male reproductive behavior, what you get is an infection of a male means that it cannot mate with an uninfected female. If it does, a sperm cannot fertilize the embryo. It doesn't work. However, if a female is infected, her behavior changes very differently. She becomes hyper promiscuous, mating more, laying more eggs, and she will even mate with other species that she would normally not mate with. And it makes sense in the perspective of the parasite, right? Because the more that you can mate, the more that you can spread that parasite further and further. So presumably there's a behavioral change that this parasite is driving. The parasite also causes there to be holes in the brain of the animal. So it's not just that the parasite will make them feel special. There are drastic long-term negative effects on the brains of the animals that are infected. They're not going to live a full lifespan, presumably because of all these tiny holes within the brains. So Wolbachia normally infects 2 out of every 5 insect species, right? 2 out of every 5 insect species, not just fruit fly, but every species of insect on the earth. 2 out of 5 are infected by Wolbachia. Will that increase or decrease? It'll increase the parasite population. Butter knives. It presumably will also increase the population, although there are side effects to laying so many eggs and having such a very fast reproductive activity. It causes there sometimes to be smaller embryos, which can be fitness disadvantages, which means they might not survive. So it's not a guarantee, but it should drive up the population. So Bite Me Medusa, there are viable offspring from the cross-species fruit fly mating if the animals are closely related. So Drosophila simulans, Melanogaster, Seychellia, and Merciana are sister species. They're all very closely related. And the animals, if there's interbreeding between the species, you will produce a F1, meaning a next generation. However, those individuals are sterile. So they're viable insofar as you can have them survive, but then they're dead, right? Because they're not going to be able to produce anything else. What are the implications of reduced fruit? You mean butter knives if you reduce the population of fruit flies overall? So usually it's an ecological breakdown. What you'll end up happening is normally, exactly, it'll be a chain reaction with the fruit flies, because they, are required as substrates for animals. Other things are eating them, breakdown products, breaking down rotting products, etc. Right, are all gonna be takeaways and negatives if you dip the population too low. Um, the Wolbachia is already gonna be worldwide, so it's not gonna cause, we don't think, any kind of additional population tank. Um, but a reason why it's important to study this is because there's a lot of behavioral changes that the bacteria causes. There's also how transmission happens of the bacteria, right? So we already know that mosquitoes carry things like malaria, dengue, Zika, right? They kill people all over the world in the number of millions. And so it's very important to figure out how you can control these animals and not have them spread that disease states. And so it turns out, we'll talk about this in a little bit, but Wolbachia can actually prevent some of these diseases from colonizing a mosquito. But what's important is to understand how else Wolbachia functions too. You don't want to necessarily just go out and infect a bunch of mosquitoes with Wolbachia and then just have it go, because you could have a lot of side effects to it, right? And so in females, like we said, some of their behaviors end up changing, and they end up wanting to mate with more and more individuals that they had not mated with before, and in terms of number and in terms of species as well. Is it okay to intervene? So I don't know if we would need to save them, butter knives, the fruit flies, because their populations are still fine. But here, the intervention is going to be, do you take Wolbachia and infect mosquitoes with Wolbachia and lower the chance of spreading dengue or Zika or something like that? And that's already actually been done. There have been field tests in Australia to infect mosquitoes. Hold, please. Paper. Wrong current biology one. So it is titled Wolbachia-mediated reduction in the glutamate receptor. mGluR promotes female promiscuity and bacterial spread. And here's the nice graphical abstract as well. So you have the infected on one side, the uninfected on the other side. When they're uninfected, the females are not as receptive to males of other species. They're also not receptive to males of their own species if they've been attempted to be multiply courted, right? And then what you see is if you infect them, they're all of a sudden much more receptive. In the paper, the researchers looked at different protein levels in the brains of the larvae. And they found this OPB, which is an odorant binding protein, shoots up, and these glutamatergic factors go down. And so what they end up looking for is, I thought this was a very elegant way of doing things, and I think Mrs. Spites will very much enjoy this. This protein, this odorant binding protein 99B, is significantly increased in the brains of the larvae. And that is leading to decreases in this glutamate receptor. So what happens? You cause mutations. All right. So while the choo-choo is going on, we'll just continue on a little bit here. What I thought was really neat was that they isolated these genetic factors, right? And what they find is this mGluR and this TFAP2 decrease in these animals. So what they did was to see if this is important. They actually genetically decrease those same genes in the fruit fly females, and they find the same behavioral effects. So these are now in uninfected females. If you get rid of those two target genes that they found that went down, the females are now all of a sudden very receptive, and they start mating at the same level as if they were infected with Wolbachia. So it's one of those really, really clean-cut experiments where here is this genetic change that we find, And after we find it, we validate it by getting rid of that gene. And lo and behold, the behavior is the exact same. And then they also saw, remember, this odor binding protein goes up. So what they do is, okay, let's get rid of the odor binding protein in Wolbachia infected females so that it can't go up in gene expression. And what do they find? Those flies' receptivity doesn't change. Pretty sure I've been tickled. So yeah. All right. So let's go over this study in a little bit more detail. What were the big takeaway messages? So we know that fruit flies have brain regions that are responsible for sensing and the decision making, right? So there is the antennal lobe that senses odorants, visual system that senses visual components, central body of the brain that makes decisions. Now they're wanting to figure out in the paper where Wolbachia are in those regions and how they're acting in those regions. So this is, again, the first time that they did this on fruit fly female brains. Normally, they've only looked at differences between infected and uninfected brains of males. But here, we talked about this before, y'all, is that this shouldn't have been the first time that they tested and looked at real differences between females, but it was. Normally, they're only looking at males for Wolbachia studies, especially this genetic transcriptions. And that's just because that's what all the literature has been focused on, and it's just been easier because they've identified something like, here is a behavior that is easy for us to characterize, and why go to something else? But this time, they actually did a compare and contrast in infected and uninfected female fly brains. What they did is they did a proteomics approach, like we said. So they didn't just look at genetics. They didn't just look at to see what RNA molecules are changing with and without infection. They looked at proteins and protein amounts. And this is the first time this has also been used for Wolbachia infected fruit fly females. What they find is the protein levels between infected and uninfected are very, very different. We'll see the graphs in a minute, but they found a total of 170 different proteins that changed. Some of them increased with respect to uninfected, some of them decreased. And those are right here. The odor binding protein 99B, mGluR, TTAFP-2. So those are like the big hitting targets that they identified here as well. They also found that there were 700 Wolbachia specific proteins in the brains of these fruit flies that were infected. So not only are their proteins changing, but now they have the bacteria, like the Wolbachia bacteria, actually identified in the brains of these animals. Makes sense that it would be there because they're infected with Wolbachia, but still, it's a really high level of that bacteria that's just hanging out there. And so they used, actually, y'all have had questions about this, but there was an AI program that they used called AlphaFold. And this highlighted what the protein structures might be looking like, as well as what are the most abundant proteins out of that list that they identified that could be the most interesting. And so what they also find is that Wolbachia also produce proteins that have nothing to do with these kinds of changes in behavior, but could also be maybe doing some other kind of behavior. And so the Wolbachia helps make some nutrients for its host and gives the animals an advantage over ones that are not infected in terms of that level of survivability. In terms of the detriment, they didn't go into so much as like figuring out what exactly the cell type is that's being affected. They looked at whole body mutants. So there could have been a little bit, more, but still, I think it was really, really incredible. On their figures are also nicely clean cut. They draw out, Falcon, what exactly they did. Collect new flies, isolate the males, do the mating, 7 days, count adult. Like a very nice lineup for how exactly the behavior, like what the animals are that they collect, seeing what hatches. And the reason this is important is because you can see if there's a deficiency in being infected or not with respect to reproductive behavior. And here is where they did the copulation recordings for about an hour each, and just see like if there's more or less in mating when infected. And they were able to find that, yeah, there is more. It's like those little pink circles being circled, those are fruit flies in there, and they are all in a mating. They're all mating right now. Which again is like, all right, it's a very easy, clean thing to see. Like, yes, they're mating. Cool. And here, again, for our friend Mr. Spites, they did immunohistochemistry, looking at the brains of infected and uninfected individuals, and at different parts in the brain as well. So here is your infected larval brains. Here is your uninfected larval brain. Their central brain. This is of a larva. So yes, here you can actually look and see in the cells. So the white here is DNA, and the green is the bacteria. You can see that there's bacteria in the cells of the central brain and in the ventral nerve cord in an infected individual, but there's not any real signal in the uninfected individual, which is as you would predict. So it's just a way of validating what they're seeing here. And then here is the full-on brain of the adult. They could be Falcone. They could be. Look at this is the adult brain. Streaming Falcon. This is a classic antibody stain called NC82. It marks neuropils or neurological structures with bundles of neurons. And see, they can even show us the mushroom body is here. There's the ellipsoid body. There's the fan-shaped body. There's the antennal lobe. There's the subesophageal ganglion. So you can see throughout the picture these various structures, and now you're just looking and seeing where the green signal is located. So the green here is the bacteria. Where is it hanging out? In uninfected individuals, there's some random color, but it's not co-localized with the DNA. So it's what's called background effects. Like, see, there's a little haze of green, but it's not a punctate strong spot. And then when you compare it to this, there is actual spot, like a physical, I can make out this is a spot bit of color, rather just a haze of color. The haze is just what's called background information. It's a background stain. It's not actually conveying you any extra information. All that's telling you is that you put your stain on, and there's a little bit of color diffusion, and that's it. These puncta are real signals. And so you can actually see the bacterium with these real signals. And here, this is the ellipsoid body. That's part of locomotor behavior, where you can see signal. Antennal lobe also makes sense that you would see a lot of signal. That's where the animals are able to detect odor. Right here in the overlap as well. Subesophageal ganglion, feeding behavior. The mushroom body, they see this patterning too. And then this bottom one is the fan-shaped body, which is integrating visual information, like visual learning and memory, is integrated at this spot and then goes out to the mushroom body, which is the other learning and memory center. And so they see this like reliable pattern of where the bacteria like to hang out with the infected animals. And then they did the sequencing, I guess the proteomics here. And they got this graph here, named after our friend Volcano Doc. This is a beautiful volcano plot. It looks like a volcano erupting, is why it's called that. And the reason that it's looking like this and having this erupting like behavior is giving you differences in gene expression, or protein expression in this case. The ones that are in black are not statistically significant. So those are ones that you're not going to be looking at. And that doesn't mean that they're not important. There is a lot of data suggesting that things that are not statistically significant, those changing could have a huge effect on the biology of the animal, but you need a way to filter this, right? If you don't filter your list, you can't test 170 different things, right? How are you going to test your favorite things? So in red are the proteins that have gone up. In Wolbachia infected brains, in blue are the proteins that have gone down. And so what you want is if it's higher and further out like this, and that's a really strong candidate, as well as these. So further out, away from the center of the plot, gives us statistical significance in terms of the fold change. So the order of magnitude difference is greater if you're further away from the 0. So if you go this way on the x-axis, that means it's a bigger change. And then the greatness in change is illustrated by the height of the graph. Now, I think it's a really, really elegant system for them to be able to do this. And then they would actually be able to do, that you'd want to do, is to do single cell sequencing, where you can actually figure out individually what the neurons are that are being affected. And then beyond from that, they also find Wolbachia proteins, the bacterial proteins in the brains of the flies. And here, this is the model that they have. And make it giant. Here we go. This is the model, and I really like this. So, Mel, this is like the artist side of me coming out. I love papers that have summary figures. That's like, here is a nice, succinct drawing of everything that mattered in what we just told you about. And that's probably just me, but, you know, that's neither here nor there. So you have, sure, the uninfected individuals here. As their brain develops, here are these transmembrane mGluRs and their neurons and the odor and binding proteins. So there's some signaling in the GluR, and there's some odor transport happening, and there's some decision making in the central complex of the brain. And so then the brain normally senses, has some perception, has a mating decision to make, and then you get uninfected progeny. Right? Straightforward. There's 3 proteins. They're doing their thing. There's this sensory input. The females are like, do I like you, male? Yes or no. If I do like you, we mate. If I don't like you, we don't mate. You might be like, Balint, that seems very niche and specific. But that's actually what the behavior is that they're looking at. And then the change is, in this instance now, you have Wolbachia, shown in green. You have it in the central part of the brain, as well as the ventral nerve cord, right? And now, they're having changes in that sensory perception. It looks like there's increases in glutamate signaling. There's increases in this odorant binding protein transport, right? And then there's also changes in behavioral decision making. And the reason we think that is this lowered level of the TFAP2. Yep. So less of the yellows, less of this mGluR, less of this TFAP2, more of the odorant binding protein. And so then now that's leading to a sensory perception overload, a mating decision overload, and then the infected, you can see it, if the circle is not filled, it's uninfected. If it's filled, it's infected. The male mates with the infected female, and now all the progeny are infected. Right? Like, how cool is this system?